I know we are all busy, and I have no intention of wasting anybody's time. So, without wasting time, you are welcome to class. This is CRP305, Crop Genetics and Breeding, and I am your facilitator. My name is ALAWODE. So, I will be taking you through this course. And like the name implies, plant breeding and genetics is just talking about the genetic makeup of the plants. Why is it that one plant differs from the other, even though they are from the same parents? Why is it that when you plant something, you expect to get one result, you get another different result? These and many more are the things that you will know as we go on with this course. So my email address is yalawode@noun.edu.ng. Feel free to ask any question that you don't understand. I'll try and make this class as interactive as possible because I don't want to be the only one talking. So I'll teach, and I want to know if you are following. So I'll ask questions also. Okay. Without wasting much time, let's move on. The definition of something. If you don't know the definition of something, then abuse is inevitable. So, what is the definition of crop breeding? And what is the purpose? The crop breeding is simply an art and science. It's an art because it's not everybody that can do it. You have to be trained, like the training you're going through now, like an introduction. It involves so many things. It's an art because you have to train for it. And it is a science also. Because there are a lot of scientific activities to it. So all you are doing in crop breeding is you are changing and improving plants genetically. So the base of every human being and plant is their genetics. The genetics that makes you different from your sibling. The DNA, nobody can share it in the whole world. You are peculiar to your DNA. So also with plants, plants have their DNA. That is what makes them different from every other plant. Behave is different from the way your brother or your sister will behave because of the genetic makeup that is inside you. So also plants, the behavior of plants on the field as a result of the genes inside them mixed with the environmental factors. So what you can see as a result of genetic makeup and the environment. So crop breeding is an art and science of changing and improving plants genetically. And this art of breeding lies in the ability of a crop or a breeder to observe plant differences. As a plant breeder, you cannot move with your eyes closed. You have to observe. Do an observation. When you go to the field, as a plant breeder, you know what you are looking for. You observe, you know, which, depending on whatever criteria you are looking for, the yield, direction of the yield, it is the number of pods for plants that produce pods, like this, for instance, cowpea. They produce pods, and you know that the more the pods, the more the yield of that cowpea. So you have to observe. You have to observe the plant differences, which may have economic value, like in a cowpea that I gave, for instance, the economic value of the cowpea is the pod. If the cowpea is growing and is growing green leaves and everything is fresh, just growing, does not produce pod, you cannot say at the end of the day that you are successful in planting that cowpea, because it has to produce pod and produce plenty of it. But if it is vegetable, for instance, and you see vegetable growing, green leaves, big leaves, then you know that because the economic yield of the vegetable is the leaf, then you as a farmer is happy. So that is the definition of crop breeding. Moving on, what is the purpose? Why all these jargons? Why are we talking about plant breeding? What is it important for? Why can't we just eat our crops and forget about this plant breeding of a crop? So the primary purpose of plant breeding is to develop varieties that are, one, efficient in the use of plant nutrients. When you are eating, you are not just eating because it is sweet. Yes, it is part of it. When you eat a food, it has to be palatable, it has to be sweet. But most importantly, what is the nutritional value that you are getting from that food that you are eating? So you develop varieties that are high in nutritional value. And the only way you can do that is through crop breeding. Because the truth is that you cannot get all the nutritional value that you need from one crop. Well, you can increase the nutrients in the plant through crop breeding. So the number one reason is to efficiently make use of plant nutrients. Then two, to give greatest return of products by, you want, as a farmer, you are after yield because you want to make money. When you have crops that are high yielding, you'll be able to sell them and get your money from them. Also, to improve natural and nutritional quality, that's the vitamin and minerals of the crop. And adapted, you need varieties that are adaptable to all seasons, all environments. You want to be able to plant your crop in the north and to do well, take it to the south, do well, take it to the east, do well. You want an all-round crop. That is another primary purpose. And you want your crop varieties to be able to withstand conditions, harsh conditions of cold, heat, drought, and flooding. You want a crop that will be able to stand the test of time. And you also want crops that will be resistant to and tolerant to plant and diseases. Because if you do everything and insects come and eat everything on the crop, at the end of the day, it steals you. So you want to develop varieties that will either be resistant. These plant pests and diseases will not be able to go near them, or tolerant. Even if they come near them, they won't be able to feed them. Just be tolerating each other. That's what tolerance means. So these are the primary reasons, primary purposes of crop breeding. Okay. Having said that, we want to talk about biological variation. When you hear anything variation, variation just means difference. So biological variation are plant differences. Biological variation is just a scientific name. What it means is difference in plants. Why is it that when you plant one, you know, a cob of corn, there are so many corn on one cob. When you plant that same cob, all the crops on all the seeds of corn on that cob, when you plant them on the field, you will see different results. That is what we call biological variation. So plant traits like the yield, the height, you see some plants, they are the same, or it's the same corn, but some will be short, some will be tall. Some will produce one cob, some will produce two. So those are differences, plant differences, and this is what the plant breeders use. This is what they manipulate to get new varieties crops. Okay, moving on, we want to talk about the causes of biological variation. What are the causes? We have been talking since we defined crop breeding, we said the purposes of crop breeding, we defined biological variation too. So we want to talk about what causes this biological variation, and there are three causes, like you can see, listed here, three of them. The first one is the genetics, and that is the genetic constitution or genetic makeup of an individual plant. Okay? And that genetics is usually passed from the parent to the offspring, generation to generation. That is why when you see a maize plant and they tell you that what plant is this, you won't say, this looks like mango. No. Corn is corn all over the world. Go to America, you see corn, you know that this is corn. Go to India, you see corn, you know this is corn. Now, there may be a bit of variation because of the weather, but the appearance, basically, you know, it grows like grass. You know that the leaves are not wide like a cowpea, it is thin. You know, so the genetics, that is what is passed from generation to from the parents to the offspring. That is why you as a child look like your parents. It's either you look like your mom or look like your father. And sometimes you may not even look like both of them, but somebody will now say, this person looks like grandma in the village. Oh, still the genes that are still there. They are passed from generation to generation, from parents to offspring. That's talking about the genetics. And also the environment causes biological variation. An environment is simply where the plant is planted. Okay? Where you grow up determines how you behave. If you grow up if you see a child that grows up in the suburb, somebody that grows up in Ajegule, and somebody that grows up in Lekki, there's always a difference. So also with plants. The environment where you plant your crops is also important. It brings about variation. That is why you see mango in the south, in Ibadan. When you bring mango, okay, let us talk about the common Ogbomoso mango. If you take Ogbomoso mango, and then you take mango from Benue State and you put it beside each other, you'll be wondering that is this mango? Because the Ogbomoso mango is smaller, while the one in Benue is very, very big. Very big. In fact, you can share that, you can get 4 of the Ogbomoso mango inside the one in Benue. So, because of the environmental factor, the environment where they grew is different. So, in that environment where the plant is planted, there may be availability or lack of water. There's lack of water. Some plants have a way of adjusting to drought conditions. You see that their fruits will be smaller. If at all they produce fruit, it will be smaller. But if you take the seed of that same plant and go and plant it where there's water, by the time you see the fruit, it will be bigger because of the environment. So the environment may lack water, it may be available with water, it may lack nutrients, it may be available with nutrients, it may lack even insects and diseases. If you see a crop that insect and disease has not finished, you will see the crop looking fine. If you take it near one that the insect and disease have already finished, you will see the difference. So the genetics and the environment have a way of affecting biological variation or differences in plants. Also, the third cause is the interaction. So we have the genetics on its own, we have the environment on its own, then we have the interaction of between genetics and environment. The interaction. So the differences you see in plants is a function of the genetics, environment, and the interaction between genetics and environment. So these are the three causes of biological variation. So let us put it in a mathematical form now. There's nothing difficult about it, because sometimes when students see mathematics, they begin to shake. There is nothing difficult about it. You can see it in equation form here. This VP is equal to VG plus VE plus VGE. And the meaning of V in this context is variation, variance of variation. Variance, because we are talking mathematics, variation is the difference. So P here, this VP that you can see, let me use my pointer. This VP that you can see is the phenotype. Phenotype means the physical appearance. What you see, you as Oluwatoyin, Teniye Oluwa, you have a physical appearance. When you appear somewhere, everybody, they will not call you Shayo, or they won't call you Tunde. They will call you Oluwatoyin because you have a physical appearance. So that your physical appearance is your phenotype, how you look. So this VP is how you look, how the plant looks. So the plant, the appearance of the plant is equal to, that means it is as a result of VG, which is the genotype or the genetic constitution of the plant. We talked about it before. Genetics plus VE, which is the variation in the environment, like we talked before, plus VGE. Now this GE is interaction between the genotype and the environment. So you as a person, your appearance is due to your genetics plus the environment that you grew up, plus the interaction of the genotype and the environment. So also the plant. Any plant you see, the physical appearance of the plant is as a result of the genetics that the plant is carrying inside. That one you cannot see. You cannot see genes. It is located on DNA, so you cannot see them with your physical eyes. But you can see the phenotype. So this phenotype is genotype, variation in genotype, plus the environmental variation and the interaction of the G by E. So the environment here is where the plant is planted, like I explained. Now, crossing, because we have talked about what causes biological variation. We know the equation now. So let's move on and talk about crossing. Crossing is simply transferring pollen or anther. The pollen and the anther are the male part of the plant. So you are transferring the pollen to the stigma of the same plant or another plant by man or by nature. Sometimes when you want to get results as a plant breeder, you have to do pollination by yourself. You don't have to leave pollination. Of course, plants will surely get pollinated anyhow. But if you want to monitor it yourself, you don't want to leave it to chance. You don't want to leave it to wind or to water or to be waiting for all that process. You do the crossing by yourself. And it's simply getting a very, very sterile instrument like your forceps. In the case of plants that have because the pollen is usually very tiny. Sometimes they are in powdery form. So you have to be careful when you are doing the crossing. So you are transferring the pollen from the anther of the plant to the stigma of the same plant. Okay. And crossing is a phenomenon where breeders create variation in plants. You can also create variation by crossing. Okay. Now, we have to go cellular. We have to go cellular. Musa Farouk, it's like I've seen this name before. You're welcome. Good morning, Musa.
Good morning, Ma. You took us the AGR 201.
Good. So how was it?
Fine. It was okay. Awesome. Okay. Thank you.
I just remembered I've seen this name before. All right, so you're welcome. Congratulations then. So
Okay, thank you,
you have moved on to 300 level. That's good.
Yes.
Okay, but you came late. What's the reason? Starts by 10 o'clock.
I just arrived in my working place, ma.
Okay. Next week. So we have already discussed some things before you came, and the link will be available. You can go back and go and listen to it. We started with definition of crop breeding, and then we talked about variation. Talked about the purpose of crop breeding at first. Then we talked about biological variation, causes of biological variation, and now we are moving on to the cellular level. Because when you are talking genetics, most of the time it occurs at the cellular level. That is why many people think it's difficult. It's not an easy process. I won't deceive you. But I don't want to make you scared, because there is nothing to it. Once you master it, most of the things here are abstract because they are at cellular level. They are abstract, but I will try as much as possible to, get things on the internet so that you can see how these things are done. So, plant cell now, because we are going deeper, so we have to go to cellular level. Cells vary widely in their structure and function. Okay, so cell division, because that is where all these genetics and all this take place in the cell. And cell division is a process through which the cells reproduce themselves. Because all these things we are doing, some people, we sleep, we grow. When we sleep, we grow. We don't know how, we just know that we are growing as human beings, as plants too. Plants, you just, you plant a seed now, and you come back 2 weeks after, you see that the seed has grown. How? It is through cell division. So your cells keep dividing and multiplying, dividing and multiplying. That is how growth takes place. So cell division is the process through which cells reproduce themselves, and multicellular organisms grow through mitosis or meiosis. Those are the cell division processes. So when a cell divides, first of all, forms what is called a daughter cell, a small cell. Of course, when the mother cell divides, it becomes the daughter cell. So following further division, these daughter cells too grow, and then they become mother cells. They divide again until the plant dies. Cell division does not stop. So cell division is the process of duplication or multiplication. So you keep dividing and growing, dividing and growing till that plant stops growing and eventually dies. Okay, moving on. And talk about gametogenesis. Gametogenesis is simply the process in which gametes are formed. Gametes Gametogenesis is from the word gametes. Gametes are the organs that are responsible for this cell division. So we have the male gametes, which are the male organs, and the female gametes, which are the female organs. So gametogenesis is just a process by which these gametes are formed. So, and this gametogenesis is a biological process by which haploid male and female gametes are formed. I'm just trying to explain it further. It occurs both in plants and animals. It occurs in plants and animals. And in higher plants, there are two stages of this gametogenesis. We have the sporogenesis. Sporogenesis, formation of male gametes, while the gametogenesis is formation of female gametes. So we have two stages, the sporogenesis and gametogenesis. Sporogenesis is formation of spores, while gametogenesis is formation of gametes. That is clear. And the gametogenesis leading to formation of male gametes are in two stages. So we first of all talked in the previous class that gametogenesis is a process by which gametes are formed, of course, in males, in plants and animals. So there are two stages. We have sporogenesis, which is the formation of spores, and the gametogenesis, which is the formation of gametes. So we'll go further to talk about the gametogenesis side. And this gametogenesis leads to the formation of male gametes in two stages. There are two stages. We have the microsporogenesis and microgametogenesis. Just follow me. These words can sound big in English or whatever, science, but you don't have to be scared. They are simple to know. Micro means tiny. Sporogenesis, spores, formation. Genesis means formation. So microsporogenesis is a small spore formation. Microgametogenesis is micro for small, gameto for gametes. Genesis is formation. So these are the things you should know. And the androecium of the plant is made up of the stamen, the filaments, and anthers. If you want to enjoy this course very well, you have to know the parts of a flowering plant. I expect you to know that by now. It's something you can look up. When you know the parts of a flowering plant, you will know where the stamen is, the filament, and the anthers. These are male organs. Okay, this androecium is made of this. The stamen is the male part of the plant. Filament, that's what bears the anther, the pollen grain. Okay, so these pollen grains carry male gametes and are contained in the anthers. And these anthers anatomically divide. They divide into lobes, which are further divided into chambers called the microsporangia. So, like I said, it's a process. I'm just explaining it so that you will understand where we are coming from. So when I later in the course, you will not be surprised and be wondering that what am I saying? So I have to introduce you to some things so that you will know it. You not necessarily come out in the exam or anything, or you have to know it. So you want to know the process of microsporogenesis, microgametogenesis, and the microsporogenesis, the pollen grains that carry the male gametes, they are found in the anther. So this anther now is the one that will divide into lobes. And these lobes will further divide into chambers. So these chambers are the ones that are called the microsporangia. And the pollens are also, they are present in this microsporangia. You can see why this process is important, because it's the pollen that we need for fertilization. So it's these pollens that are present in the microsporangia. And the microspore mother cell is diploid in nature. It divides meiotically. You know, when we were talking about cell division, we said there are two types. We have the meiosis and the mitosis. So divides to form haploid microspores of pollen. This whole process that we have been talking about is what is called microsporogenesis. So you can see that the first thing is the formation of the pollen, which can be found in the anther. And these lobes, they divide to form the microsporangia. It's inside this microsporangia that you find the pollen grains. And this microspore mother cell will divide meiotically to form these microspores or pollen. This whole process of pollen formation is what is called microsporogenesis. In the case of the microgametogenesis, the pollen grain is the one that divides into two halves. And the small nucleus, that's two halves. That's the small generative nucleus and the large generative vegetative nucleus. So pollen grain divides into two, the small generative nucleus and the large vegetative nucleus. The generative cell gives rise to two male nuclei, whereas the vegetative part gives rise to the pollen tube. So this process is the one that explains the formation of the microgametogenesis. Like I told you, most of these things occur in the nucleus. They are not things that you can see with your eyes, but we have pictorial illustrations that you can check. It's all over the internet. Just type microgametogenesis there. It brings up pictures you can see and relate it to these notes that I've already given you. See, the first one we talked about before was microsporogenesis. That's the formation of the pollen grain. Now in the microgametogenesis is the pollen grain now that divides into two. You have the generative nucleus and the larger vegetative nucleus. So this generative nucleus is the one that will give rise to two male nuclei, while the vegetative part of it is the one that will give rise to the pollen tube. So this explains in a simple way the process of microgametogenesis. So that is for the male part. Now, we want to talk about gametogenesis of the female gametes. Okay, and this one too occurs in two forms, two stages. Like we talked about the male formation of male gametes in two stages, which is the microsporogenesis and microsporogenesis and microgametogenesis. We want to talk about the female ones too. Those ones occur in two stages, which is the megasporogenesis and megagametogenesis. Mega because they are larger. Micro means tiny for the males. Mega is for the females and they are larger in size compared to the males. That's why it's called mega. So it should not confuse you. And in the formation of the megaspore or megasporogenesis, the ovules are the ones responsible. You know, we are talking females now. We cannot talk of ovule in male. There's no ovule there. The ovules are present in the ovary in multiple loads. And a cell in the ovule is differentiated or split, divided into a megaspore mother cell. This megaspore mother cell is diploid in nature, it's two. So this megaspore mother cell will now undergo meiosis to form 4 haploid megaspores. 3 out of these megaspores, out of the 4, will degenerate, we die off. There's only 1 that will remain, and that one is left in the ovum. So this process of a cell differentiating into megaspore mother cell, and the megaspore mother cell forming 4 haploid megaspores, and 3 of these haploid megaspores will degenerate, 1 remaining. This whole process is what is called megasporogenesis. In the case of megagametogenesis, this megaspore that was now formed, that one that is remaining, will divide again to form 8 nuclei. So out of these 8, 6 of them will migrate to the opposite poles, while 2 will remain in the center. So the one that remains in the center is what we know as polar nuclei. And this polar nuclei will fuse to form the secondary nuclei. The megaspore mother cell now matures into the embryo sac. That is where the female part is. So this entire process is what is called megagametogenesis. Please, I want you to go and do it better. This is a picture that I have here of the megasporogenesis process. You can see the megaspore mother cell here. Divide into 2 haploid, further divide into 4 tetrad. So out of these 4, 3 of them will disintegrate to remain 1. That is what is here. And this one will divide again into 2, from 2 to 4, from 4. That is how it will be till it forms the embryo sac. So these are the processes that are involved in cell formation. After talking about the gamete formation, we want to talk about fertilization. We have talked about male gametes, we have talked about female gametes. So fertilization in plants is the union of this male and female gametes to restore the chromosome number of 2n or diploid. So the union of male gametes and female gametes is what we know as fertilization. And in plants, this fertilization occurs in sexual reproduction, and it occurs after pollination and germination. Okay, fertilization occurs when you have done your pollination. It will give rise to a seed, then it will germinate. Okay, what I'm trying to say now is that you have the male gamete, you have the female gamete, they come together in a sexual way for plants, and then form a seed. That seed will germinate to become the plant. So fertilization is the fusion, like I said, of the male gametes, which is the pollen, and the female gametes, which is the ovum, to form a zygote. And that zygote is diploid in nature. It is a physical-chemical process, and it occurs after pollination of the carpel. So the process of fertilization, first one is pollination, and this takes place immediately before anthesis. When you grow a seed, like cowpea, for example, cowpea will continue to grow vegetatively. See leaves growing all over the place and everything. After like 2 months, in some cases, it starts to flower. Once that plant starts to flower, then it is ready for fertilization. Pollination is ready to take place. So when pollination takes place, then it will now set and form seed. And all this will happen before anthesis. Anthesis is like the end of the process of a crop. So pollination takes place, and the seed is formed before the plant now dies. And within after pollination, then pollination germination, within 2 to 3 minutes, the pollen left on the stigma start to germinate. Like I said, after pollination, then germination will take place, germinate to grow. The pollen will come onto the stigma. Like I said, sometimes you as a plant breeder has to do it. You have to do the pollination process. Otherwise, you leave it to nature. The wind, when wind blows, it will carry pollen all over the whole place and carry it to a stigma, and pollination will take place. Sometimes it's water, sometimes animals, when they eat some plants, they excrete it, they excrete pollen all over the whole place. So pollen flies around, and once the pollen has come on the stigma, fertilization will take place. After fertilization, then division process will take place to form the zygote. So that is about the fertilization process. Now we have been talking about nuclei, we have been talking about this and that. Where can we find it? We can find it inside the plant cell. And what is a plant cell? A plant cell is the smallest unit of life. All living organisms are composed of the basic units, which range from simple unicellular structure, bacteria, that's for the structure for organisms that have only one cell. Call them unicellular. Example is bacteria, protozoa, and to complex structures of plants and animals. We have billions and billions of cells inside our body. Billions. Plants also, they have billions of cells in their body. And this is what a cell, a plant cell looks like. You have the cellular cell wall, you have the cell membrane, you have the Golgi apparatus, you have the vacuole, chloroplast, which is responsible for giving the plant its color. You have the mitochondria, that's where the energy, that's the energy package of the cell. You have the cytoplasm. Inside the cytoplasm, you have every other cell activity going on. Then you have the nucleus here. It's inside this nucleus that you find the DNA. Inside the nucleus that you find the DNA of that plant. Okay, we have the nucleolus at the center of the nucleus. We have the ribosomes. All these parts, you should know by now what it can be used for. We have started doing all these things from secondary school. Plant cell and animal cell, differences between plant cell, differences between animal cell, what is useful. I'm not going to go into that now. You are 300 level students. I want to believe that you are mature, to be able to know what these parts of the plant cell is used for. So this is what a plant cell looks like. Of course, this has been magnified. In the real essence of it, you can't even see it with your naked eyes, but when under the microscope, this is what you're going to see in the plant cell. Okay. Cell division, like I talked about before, is the most fundamental part of the plant cell, of the cell division, and is replication of the DNA. And two processes are involved in cell division. We have the mitosis, which is the nuclear division, and the cytokinesis, which is the changes in cytoplasm. So two processes involved in cell division is the mitosis, which is the nuclear division, and the cytokinesis, which is the change in cytoplasm. For mitosis, new cells are formed, and these cells are identical. Okay? They retain the chromosome number and heredity of every cell. So in man, for example, man has 46 chromosomes. And after the mitotic division, cells will still have 46 chromosomes. After mitosis, in man, cells will still remain 26. I've talked about gametogenesis and fertilization. And for example, now, these are examples. Okay, let's see how gametogenesis works. Like I said, man has 46 chromosomes, and these chromosomes are denoted by 2N, or diploid. So in these 46 chromosomes, 44 are autosomal cells, while only 2 out of these 46 are sex chromosomes. So autosomal cells are 44, only 2 are sex chromosomes. And during gametogenesis, the chromosome number will be reduced. Meiosis is a reduction reaction. While mitosis, like I said, the cells will still be intact, but for meiosis, to be halved. Okay. So during gametogenesis, the chromosomes should be reduced to half. So you have 23 chromosomes, which is haploid now, and is denoted with N. For the diploid, is 46, it's denoted with 2N. Or if it is haploid, it is denoted with N and it's 23. In maize has 20 chromosomes, which is denoted as 2N, called diploid. During gametogenesis, the number will be reduced to 10 or N. So you can just play around with different organisms, look at their chromosome numbers, and try and know, and find out what happens when the cells divide, so that you can just have an idea of what happens. And like I said, diploid is a complete chromosome complement denoted by 2N. Haploid is half, which is denoted by N. This reduction is caused by meiosis, and meiosis therefore is the process by which chromosomes have been halved. The process of, when they are halved, they don't remain like that permanently. When they join again together, they become full. But the process will first of all divide it, reduce it into half. Then mitosis will now set in and complete it again. That's what happens in cell division. We have characteristics between mitosis and meiosis. We have a list of them here. I think I have 1, 2, 3, 4, 5, 6 points. Differences between mitosis and meiosis. In mitosis, 2 products or 2 daughter cells are produced per cycle. In meiosis, 4 gametes are produced, 4 gametes. Then in mitosis, genetic content are identical, while in meiosis, the genetic content are different. They are not identical. And in the case of the chromosome number of daughter cell is the same as that of the mother cell. Okay, daughter cell and mother cell have the same chromosome number in mitosis. But in meiosis, the products are halved. Okay, the daughter cell is half of what the mother cell is. So the products of mitosis usually undergo additional mitotic division, and in meiosis cannot. In mitosis here, when a mitosis division occurs, it is followed by another mitotic division. But in the case of meiosis cannot follow meiosis again. So it will further, like I said, meiosis and then followed by meiosis. Meiosis followed by mitosis. That is how it is being done. So in mitosis, while in meiosis, it can only occur in the gametes or the gametic cells. Mitosis is associated with asexual reproduction, and meiosis is associated with sexual production. So it's important for you to know the differences between mitosis and meiosis. It's important for you to know it. So having said that, we have come to the end of today's lecture, and I hope you understood every bit of it. Like I said, if you don't understand anything, my email address is there. You can always contact me. And my turn to ask my questions. I hope you are still there. Oluwatoyin, I can't see your face. It's really Lisa's face I can see. I hope you are still there. Okay, Toyin, I have a question for you. You are still there. I want you to define crop feeding for me and tell me at least 4 primary purposes of crop breeding. At least you were in class when I was speaking. So I'd like to hear from you. Are you still there? I don't think she's there. Okay. Farouk, you were not in class when I was talking about it, but you were in class when I was talking about the causes of biological variation. So I want you to tell me 3 causes of biological variation. You have to unmute yourself.
No, I will talk, but not now. Sorry.
You were not in class?
I came when you are halfway of that.
Oh, really? Okay. Can you tell me about the gametogenesis process of formation of gametes?
Okay. Formation of gamete. Gametogenesis is the process in which the gamete is formed. And sorry, it has 2 phases. One is micro gametogenesis, and the other is a microsporogenesis, right? Okay. And this microsporogenesis is the process in which microspores are formed, and the microgametogenesis is the process of the formation of the microgametes in the of the male organ, right?
Yes, they are formed. Okay. What about for the megagametogenesis? Can you say something about that too?
Megagametogenesis is peculiar to the female organs.
Yes, female gametes. Okay. Right now, I'm expecting like 62 people in this class. Okay? And it's only 2 of you that I can see. Now, the results will come out, they'll say this one, that one. Where is everybody now? Don't you have a place where you people meet yourselves to tell yourselves that this is what is happening? You know, you should many people check Facebook now. They're on WhatsApp. So why can't they just join the class? What's the problem now? Because I need to know. I'm expecting 62 people in this class, according to what I have. I sent messages to 62 people, and I can see only 2. Things like this are not encouraging. So please, if you see your colleagues, tell them every Wednesday we have CRP305. And CRP303 is tomorrow too.
The causes of biological variation is genetics, environment, and the interaction between the two of them.
Genetics and environment. Okay. We have a mathematical representation of that.
Yes,
Okay.
that is phenotypic variation is equal to genetic variation plus
variation.
Plus
environmental variation plus the interaction between genetic
yes,
and
plus environmental variation, and then plus VGE, right?
Yeah, interaction between both.
Yes.
Okay. So, okay, that's all. It's 11 o'clock already. So, like I said, I don't like wasting anybody's time. So please just go through it again, and then we'll meet next week by God's grace, another class. Any question?
Okay, what do you have tomorrow, ma? What do we have tomorrow?
CRP303. CRP303. I don't know if you are doing it.
I'm not sure.
Yes,
I'm doing it. I'm doing it. Okay.
Tomorrow.
I have a lot of questions, but maybe I have questions, but not in this part. It's way further.
Okay. One question.
I said I have questions. But it's ahead of where we are now.
Okay, you have already read all this. Okay, ahead. When we get there, or let me know what the question. What question is it actually?
The question is this male sterility. Okay. I don't understand. Okay, let me just allow it till when we get there, so that I should not confuse others.
See, next week I'll try and hurry up too, because we have just 8 weeks, like you know, and those 8 weeks are virtually not enough. We couldn't cover most of the topics in Nigeria 201 last session because of time. So I don't want that to repeat itself here. I will try and be fast, and yeah, we want you to read most of these things on your own, so we can just come to class and just do it. So, okay, no problem. Have a nice day.
Thank you
very much.